welcome everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor, Biomodels, I'd like to welcome you to Current Preclinical Models in Oncology, Enhancing Predictivity for Translational Success. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. At this time, I'd like to introduce our presenter for today. He is Dr. Benjamin Cuffo, PhD, Principal Scientist in Oncology at Biomodels. Welcome, Dr. Cuffo. The presenter ball is yours.
Thank you, Elizabeth. I'm very happy to be here today talking with you all about current preclinical models in oncology and how we can use them in the most predictive way possible to enhance translational success. Before we begin, I'd like to tell you a little bit about Biomodels. We are a preclinical contract research organization located in Watertown, Mass, up the Charles River from Boston. Our view is not quite as nice as the picture you see here, but we are on the Charles, right in the Boston area. We were founded in 1997 out of the Brigham and Women's Hospital, also in Boston, and we specialize in highly translational models of human diseases and conditions for a wide variety of clients, ranging from startups to big pharma. Our goal is to transition drugs and biologics from concept into patients. And we've been successful in doing that, having facilitated about 43 compounds into patients for multiple indications. We have therapeutic expertise within the areas of oncology, cardiovascular disease, inflammation and autoimmunity, metabolic disorders, pulmonary disease, neurology and psychiatric disorders, fibrosis, and cancer supportive care and oral health. For the talk today, I'm going to focus on a few topics. Here's just an overview. We're going to go over the general cancer therapy development pipeline, talk about the success rates for novel oncology therapeutics, gradients of predictivity in preclinical oncology models, the advantages of emphasizing translational preclinical modeling, and the importance of recapitulating clinically relevant context. And finally, we'll talk about novel applications and opportunities in preclinical oncology models. The oncology therapeutic development pipeline, as you all may be familiar with, starts with early discovery and lead identification, mostly in an in vitro setting, before moving on to preclinical modeling to determine safety, and finally into clinical phases. We specialize in the preclinical sphere, but go well past where you might end, think about ending your preclinical modeling. The IND filing, you don't need a lot of efficacy testing, but you might want to consider doing more preclinical modeling before going into clinical phase or in parallel. So looking at the success rates historically for novel oncology therapeutics, what we do know is that these novel treatments that are both safe and effective are in high demand across all cancer types and settings, some obviously more than others. And we've continued to make extraordinary progress in our understanding of the biology of cancer. Likewise, a huge investment and effort has been made in the development of these novel anti-cancer agents. However, success rates remain low across therapeutic paradigms, with biologics being slightly better than small molecules. If you look at the table on the right, you can see that only 7.5% of oncology drugs in this table from 04 to 05 and have eventually reached approval. And that number has not increased much in the ensuing time. And novel anti-cancer therapeutics historically even have lower success rates than therapeutics developed for other indications. And if we look at oncology therapeutics versus non-oncology, in this case from 2011, we can see maybe where that difference is manifesting. So you can see in phase 3 to registration, you see a drop-off in oncology versus non-oncology. And this has to do a lot with eventual efficacy. So some of the reasons might be design and execution issues in clinical trials, difficulty in correctly matching patients to treatment. But our focus will be the historical, minimal, imperfect, and or irreproducible preclinical efficacy modeling. So how can we improve that? Currently available preclinical oncology models can now recapitulate critical underpinnings of cancer progression and response. So these models can accurately model cancer biology. They can model cancer in context. We can use knowledge of genetics or epigenetics of the cancer cells or the model systems utilized to make more translationally accurate models. We can model tumor heterogeneity. We can model the microenvironment. And we can see similar progression or dissemination of the cells from the primary tumor. And we can now model functional immune contributions as well. With these models, we can produce clinically meaningful endpoints that are going to be translatable and work to improve clinical trials. Interpretable endpoints like survival or progression-free survival, use of biomarkers to ensure targeted therapies are reaching their targets, and readouts such as circulating tumor cells, just to name a few. These models can also recapitulate clinical treatment paradigms, and I'm going to be harping on this point a lot, looking at efficacy of a novel therapy in combination with standard of care therapies, such as chemo or radiation regimens, and efficacy following clinical situations, such as resection of the primary tumor, where this is possible, and treating residual disease. Of course, not all preclinical models are created equal, and you can expect, based on the ability of the model to accurately describe the biology taking place, that they're going to have different gradients of expected predictive potential. So I've made this graphic sort of to show the gradients that you might expect from these different models. On the left, you can see in vitro assays, and on the right, all the way up to well-characterized PDX models using humanized animals in clinically recapitulative ways. So how can we use all of these currently available tools to enhance the probability of translational success? Well, we can use preclinical studies to identify the patients most likely to benefit. This is a very important point. And model in the context of the treatments that these patients are currently most likely to be getting. We can model the situations where the patients will most likely first see these therapies in terms of metastasis, resistance, dormancy, or against cancer stem cells. We can, again, recapitulate clinical context, and we can correlate with clinical observations. Now, each potential therapy is going to be unique, and it's going to be targeting a specific biology of the cancer, and maybe only effective in a certain setting. So preclinical research can not only identify a therapeutic niche for the therapy, but also identify a patient that might be most likely to benefit. To do this, we can use rational, knowledge-based, and individualized design of preclinical studies tailored specifically to the therapy. In this way, we can hope to increase our positive results and decrease false negatives. This graphic just keeps on this point. So if we use the currently available preclinical models in a smart way, rationally designed, we can take advantage of the biology to enhance our translational success. Most often this involves iterative preclinical modeling, going from one model to the other and gathering information, and then moving that into clinical endpoints, clinically relevant endpoints. We believe that investing more research and development dollars in clinically recapitulated preclinical studies prior to or in parallel with clinical trials will ultimately save money, save time, decrease false negatives, so that when you get to the point where you're not getting the result that you hoped for in your clinical trials and the therapy won't be shelved and it just hasn't been used in the proper situation for the proper patients, you're going to identify those patient populations, you're going to establish meaningful endpoints, and you'll be able to test therapies in combination with the therapies the patients will be getting. You'll be increasing confidence moving forward that you're on the right track. You'll be increasing investor confidence. And ultimately, we believe you'll increase your success rates. Now, what do we have in our toolbox for preclinical oncology models? There are many, and these can be interchanged and used in creative ways. Everything from in vitro models, and there are many, to syngeneic models, xenogenetic models, transplanting human cells into animals, metastasis models that take advantage of different stages of metastasis, humanized mice, co-implantation with human stroma or other cell types that can influence the progression of cancer, cancer stem cell models, heterogeneous PDX models, and treatment-resistant models. With a variety of different cell types, a variety of different concomitant or sequential therapies to visualize these primary endpoints, which might be telling. So just to go through a few of these, probably the most widely used historical model has been ectopic xenografts, where human cancer cells are implanted subcutaneously in the flank. And this has been the workhorse model for many years. And although it's probably the reason that the success rate is so low, it's also the most successful model because pretty much all of our successful therapies to date have seen this model at some point. So we feel that this might be, although not the most predictive model on that gradient, for some it might be a good place to start because you have cancer growing in 3 dimensions, in vivo, with your routes of dosing in a full living system available. So you can use it to sort of get a basic overview of how your tumor might respond. But it doesn't tell you a lot about ultimate survival or the many more subtle details that end up having a large impact on the progression of cancer. So we suggest this model maybe as a first pass for more comprehensive studies or to establish PK or dosing schedules with some efficacy readout. This is also used generally in limiting dilution assays to establish tumor-initiating cell populations and potentials, which I'll talk a little bit more about later. You can also use these in a little bit more creative ways, such as co-injecting human stromal cells with the tumor cells and things like that. But there are a few issues with that approach, such as the fact that these stromal cells tend to not persist very long. So how can we improve on this basic model that's sort of been historically used? Well, modeling cancer in context is going to be an improvement, even if this context is in the context of a mouse. So one of the technical obstacles in doing this is that a lot of your internal organs that you might want to be looking at are very difficult to see and measure by digital caliper. So this is where bioluminescence really comes into play and becomes a very important tool that we can use in the orthotopic setting. When you use bioluminescence, you not only get a more elegant study and a way to really visualize your efficacy in sort of real time, but you also get the added bonus of a more reliable study. When you have bioluminescent cells injected orthotopically, you can randomize the animals in in a more accurate way so that each animal that's put into each treatment arm is going to have a similar average luminescence or radiance and a similar range so that you're getting a more fair study. You can also use bioluminescence to measure progression in terms of metastases following treatment, for instance. Finally, you can easily re-isolate the cells from the animals after and look at how well you've hit your biomarkers. So it really is a great tool that we can take advantage of, especially in the orthotopic site, which is cells growing in niche, albeit different species. So Biomodels now offers a wide range of bioluminescent tumor models that we can use in orthotopic settings, and we really feel that this should be the go-to model, perhaps above the ectopic xenografts. So just to show you a little bit of data of what we can do with this model, on the top here, we've implanted these cells in the mammary fat pad. These are triple-negative breast cancer cells. And on the bottom, we've isolated for putative cancer stem cells and done the same thing and treated with the same small molecules or chemotherapies. And what we found in this case is that the mice implanted with the cancer stem cells responded quite differently than the mice implanted with the complete parent population. Now we're able to isolate the putative tumor-initiating cells or cancer stem cells in vitro or in an ex vivo way by either staining, antibody staining for cell surface markers, followed by FACS sorting, or by enriching in anchorage-independent conditions such as tumor spheres. And if we look at the in vitro, we sort of did not see what we were able to see after implanting in vivo. So you can see, again, levels of the orthotopic implantation following this enrichment can give you. So we can treat in vitro as well and then do things like tumor limiting dilution, tumor initiation analysis in animals to ascertain how many of the cancer stem cells we've actually taken care of. So there's different ways we can play around with these models depending on what we're looking at and what we want to achieve. Orthotopic xenografts, as I'm saying, are great. We think this should become a new workhorse model. However, they do have their limitations as well. So we're obviously missing elements of tumor genesis. We're missing those species-specific tumor stroma interactions, although it remains unclear how different those are from the interspecies interactions within the corresponding orthotopic sites. And we're especially missing immune contributions because these must be done in immune-compromised animals of various degrees. We're extremely excited to announce today our collaboration with Zentek to bring you bioluminescent patient-derived xenograft cell lines that can be used at orthotopic sites. Now, patient-derived xenografts are extremely useful in that they preserve tumor heterogeneity far better than traditional cell lines that have been long cultured in 2 dimensions on plastic and are thereby selected for various cells that grow in that way. Patient-derived xenograft lines tend to be much lower passage and come with a great deal of clinical patient information that can be utilized. Zentek specializes in highly characterized PDX lines, and they do a great deal of high content molecular profiling, including expression profiling, copy number profiling, miRNA, microRNA expression, and mutations analysis. They do a lot of phenotypic characterization, and they have extensive drug response profiles for standard of care, response to standards of care, and match patient data. So with these tools, we feel that this is a fantastic platform for which to do preclinical oncology studies, especially given that these have been transduced with luciferase and we can follow them, their growth at orthotopic sites in vivo. So through our collaboration with Zentek, Biomodels is pleased to now offer a panel of highly characterized bioluminescent PDX lines from a number of typesetting patient and clinical contexts. And these are an early passage, and they once they get to us, they're propagated in the orthotopic setting. So below you can just see a single line of the growth from a pancreatic bioluminescent PDX line from Zentek. But we have a poster coming at AACR and CIEORTC in Boston in November that I hope you'll visit, that shows the response of this line to some common and some standard and experimental therapies that patients with pancreatic cancer are receiving. And this has been done in a very clinically recapitulative context where we're really allowing these tumors to grow for a long time. So you're sort of modeling a late stage when most of the patients are getting diagnosed. We feel that bioluminescent PDX lines utilized in rational preclinical studies is really allowing for unparalleled visualization of tumor kinetics in vivo, and is really going to allow for highly predictive studies and identification of biomarkers that can be clinically useful. However, as I was saying before, all of these models, no matter what their expected predictive power, have some drawbacks. And in this case, I'd like to step back for a minute and talk about syngeneic orthotopic isografts or allografts, where we're implanting mouse cells instead of human cells at the orthotopic site. This allows us to model in the context of a fully immune-competent animal. This is just showing some data from a recent study we've done with an orthotopic model of glioblastoma, which is another context where there's a lot of demand, and with some radiation and standard of care. But we're able to get, in syngeneic models, an extremely high tumor take. So the advantages of these models are they do have predictive potential, even though it is 100% mouse, and the cells are grown in an accurate context, both in niche and within the proper species. We have a fully intact immune system, high tumor take, fully intact tumor stroma interactions, and tumor immune interactions. So what we're missing here is maybe some particulars of human biology, which is obviously not a small thing, but depending on what exactly we're looking at, it may be something that we can work with. We're also missing tumorigenesis here. So these types of models can be used in hematopoietic leukemia lymphoma models, for example, or in immune oncology studies, which is a very active area of cancer research. In addition to the syngeneic models, we can take advantage of genetically engineered mouse models that include transgenic knockout CRISPR'd or transduced transplanted mice that have been genetically altered in a wide range of ways. Through these models, we can induce levels of humanization into our mouse models and use them in a number of creative ways and applications. These have been extraordinarily useful historically in elucidating tumor biology, especially in tumorigenesis and progression and immune response. I'm not going to spend too much time on these because there are so many applications here. I'd like to just highlight one application which might be expressing a human antigen in a mouse setting. It can be very useful. So a special interest of mine has been metastasis and how best to model metastasis in mice. I'm going to talk a little bit about some of the metastasis models. To model the very final points of metastasis, you can implant directly at the site of metastasis. So if we know that colorectal cancer often ends up in the peritoneum, so we can inject directly in with a luciferase transduced HT29 line, for instance, in this case, and model peritoneal carcinomatosis and treat with prospective therapies. What we miss in such a model here is the earlier steps of metastasis, including local invasion, travel through the circulation, et cetera, which may be selecting for certain cells that we're not selecting when we inject a parent population in. To take a step back in the metastatic cascade, we can inject into the circulation. There are some drawbacks here where you often get cancer cells not arriving in their clinically relevant metastatic sites just due to a large bolus of cells finding its way to easily accessible capillary beds in the lungs, for example. But in many contexts, this can be an accurate model of metastasis as well that takes into account extravasation and colonization processes. So I don't think I'll read through all this, but you can read it later if you like. We can also pre-select and pre-treat or pre-treat cells to enrich, as I was talking about before. Before doing any of these metastatic models, we can use genetically engineered systems such as virally expressed hairpins to select for or push the cells into a more metastatic state, et cetera, and use models in this way to get at likely places in the metastatic cascade where your therapeutic might have biological activity. So just to give sort of a case study of how we might use these tools to put it all together, if we want to look for a novel treatment for novel therapy for treatment-resistant metastatic triple-negative breast cancer, for example, and we've done research and development in vitro to the point of a lead candidate, what type of preclinical model might be most predictive in this case? So we think maybe in this case a metastatic bioluminescent PDX line that's used in a clinically recapitulative way might be the way to go. So in this case, we would use orthotopic seeding and allow the primary tumor to grow. And then as a patient would, if possible, you would resect the primary tumor prior to the appearance of detectable metastases, and then individually enroll each animal at that randomized point that's determined by IVUS radiance, and individually enroll each animal into treatment so that each animal is starting treatment at the exact same point. So now you're treating an animal that no longer has a primary tumor, but you're treating residual or metastatic disease as you might in patients before you can necessarily see it. And as you progress, you can detect your lung metastases by IVUS, and you can combine these with standard of care therapies that the patients might be receiving. And we've actually been doing this in progress, and we're going to be presenting that data at the AACR meeting in Boston in November. So I hope you'll come see us then. Just before I sum up, I'd like to just give a nod to the humanized mouse models that can be used to study immune cancer interactions in a mouse, whereby you have an immune-compromised mouse that's been engrafted with human hematopoietic stem cells and allowed to develop a humanized immune system. And these models, we believe, will be extremely useful moving forward in immune oncology studies. And we have an active program looking at these models in conjunction with bioluminescent PDX. In summary, I'd like to say that no model is perfect. All models are facsimiles, ultimately. However, we can use currently available preclinical models in ways that are going to be more translationally predictive. And the key is really how you use the models at hand, how you use the tools at hand. And while it is true that some of the more translationally accurate models are going to be generally more expensive, we believe that ultimately this will save your company time, money. You will decrease false negatives and be more likely to put your therapy in a position where it has the greatest chance at success in clinical. So to sum up, we believe that rational use of accurate preclinical models tailored specifically to the therapy and the clinical indication can enhance the probability of translational success. With that, I'd like to say thank you very much for joining me today and listening. Please don't hesitate to contact me with any questions. I'm at your service, and I love to talk about what's coming down the line in terms of these novel therapies. I think we're at a very exciting era in cancer therapy, and I look forward to the future as well. Please visit our poster at the upcoming AACR NCI EORTC in Boston in November, and we'll also be at the AACR annual meeting, not in 2106, but next year in 2016. And thank you very much. I'm going to be taking all questions by email or by phone so that I can really devote some time to them and answer them more thoroughly. So thank you all.
Thank you very much, Dr. Capo. I'd like to say thank you so much, Dr. Capo, for your presentation. I'd like to thank Biomodels for sponsoring today's web symposium. And I'd very much like to thank those of you who came and spent some time with us. I hope you found some answers for problems that will make your work a bit easier. With that, I'd like to say thank you all so much. I hope you have a great day, and I look forward to seeing you at future CHI Global Web Symposia. Thanks, and have a great day. Bye-bye.